News release. Tuesday, March 14, 2023. Tool has the potential to assist with preterm birth, labor, management, and clinical decision making. Researchers funded by the National Institutes of Health have developed a new imaging tool called electromyontreal imaging (EMI) to create real-time, three-dimensional images and maps of contractions during labor. The non-invasive imaging technique generates new types of images and metrics that can help quantify contraction patterns, providing foundational knowledge to improve labor management, particularly for preterm birth. The small study is supported in part by National Institute of Health's Eunice Kennedy Shriver National Institute of Child Health and Human Development NICHD through its Human Placenta Project and other programs. The findings are published in Nature Communications. EMI has the potential to answer critical questions about uterine contractions and will help us better understand what occurs during pregnancy and labor, said Diana W. Bianchi, MD, NICHD Director. With additional research, the tool may potentially predict who is at risk to deliver prematurely or whose labor pattern will eventually result in the need for a cesarean section delivery. This will also help care providers evaluate whether a treatment or intervention is working. The study team, led by Yang Wang, Ph.D., and Alan Schwartz, M.D., Ph.D. at Washington University in St. Louis, and Allison Cahill, M.D. at the University of Texas at Austin, initially developed EMI using a sheep model and reported their findings in Science Translational Medicine. In the new study, the team tailored EMI for human clinical use and tested it among a group of 10 women with healthy pregnancies. Current clinical methods to measure contractions, i.e., tachydynamometry and an intrauterine pressure catheter, can only provide limited details such as contraction duration and intensity while also being invasive. EMI integrated two types of non-invasive scans, a fast anatomical MRI to obtain an image of the uterus, which can be taken during early-term pregnancy or 37 weeks gestation, and a multi-channel surface scanning electromyogram that uses sensors placed along the belly to measure contractions during labor. These data are then combined and processed into three-dimensional uterine maps, with warm colors denoting areas of the uterus that are activated earlier in a contraction, cool colors indicating areas that are activated later, and gray areas showing inactive regions. A sequence of maps is generated over time, creating a visual time-lapse that shows where contractions start, how they spread and slash or synchronize, and potential patterns that are associated with a typical pregnancy versus one with complications. EMI maps were also used to develop metrics to describe uterine contractions. The maximal activation ratio, for example, measures the total surface area of the uterus that becomes electrically active during an individual contraction. The activation curve slope measures the rate of uterine electrical activation. The fundal early activation ratio helps quantify the region that generates contractions to dilate the cervix. Results from the pilot study also bring clarity on a long-standing question on how contractions begin. EMI data suggests there is no fixed, pacemaker-like region in the uterus that initiates labor. The study team observed varied patterns of contractions and metrics among the 10 study participants, with some similarities between women who had never given birth and those who had. However, more research is needed to confirm and expand upon these observations. EMI offers new possibilities for better understanding human labor and facilitating the development of optimized, patient-specific interventions. The authors note that an EMI A contraction atlas generated from healthy pregnancies can serve as a resource to understand and diagnose preterm labor and possibly identify patients who would benefit from an induction versus those who may need a cesarean section. About the Eunice Kennedy Shriver National Institute of Child Health and Human Development and ICHD leads research and training to understand human development, improve reproductive health, enhance the lives of children and adolescents, and optimize abilities for all. For more information, visit www.nisht.gov. About the National Institutes of Health. National Institute of Health. National Institute of Health, the nation's medical research agency, includes 27 institutes and centers and is a component of the U.S. Department of Health and Human Services. National Institute of Health is the primary federal agency conducting and supporting basic, clinical, and translational medical research and is investigating the causes, treatments, and cures for both common and rare diseases. For more information about National Institute of Health and its programs, visit www.nagov. National Institute of Health turning discovery into health registered. Wang H. Wen Z. et al. Non-invasive electromyometrial imaging of human uterine maturation during term labor. Nature Communications 10.1038/S41467 23 36400 Fertizero 2023